Pinus foliage leaf. It is also known as needle. Coming to the transverse section of the needle. In a transverse section, it is circular in outline in Pinus molefila, semicircular in Pinus sylvestris, and triangular in Pinus longifolia and Pinus roxburghia. The outermost layer is epidermis, which consists of thick walled cells. It is covered by thick cuticle. Many sunken stomata are also present, and each stomata opens internally into a substomatal cavity. Below epidermis are present a few layers of thick-walled sclerenchymatous hypodermis, and it is well developed at ridges. In between the hypodermis and endodermis is present a mesophyll. The cells of mesophyll are polygonal and filled with chloroplast. And many peg-like infoldings of cellulose also arise from the inner side of the wall of mesophyll cells. A few resin-canal cells are also present in the mesophyll adjoining the hypodermis. Their number is variable, but generally they are 2 in number. Endodermis is single-layered with barrel-shaped cells possessing Casparian thickenings. Pericycle is multi-layered and consists of mainly parenchyma cells and some sclerenchyma cells forming T-shaped girder which separates 2 vascular bundles. Transfusion tissue consists of tracheidal cells. 2 conjoined collateral vascular bundles are present in the center. These are closed, but the cambium may be present in the base of the needle. Xylem lies towards the angular side and the phloem towards the convex side of the needle. Coming to reproduction. The plant body in case of Pinus is sporophytic and Pinus is monoecious, that is, the male and the female flowers are present in form of cones or stabile on the separate branches of the same plant. Many male cones are present together in form of clusters, each of which consists of many microsporophylls. The female cones consist of megasporophylls. Coming to the male cone. The main cone develops in cluster in the axil of scaly leaves on the long shoot. They replace the dwarf shoot of the long shoot, and each male cone is ovoid in shape and ranges from 1.5 to 2.5 cm in length. A male cone consists of a large number of microsporophylls arranged spirally on the cone axis. Coming to the longitudinal section of the male cone. The section is ovoid and the center is occupied by cone axis. Microsporophylls arise at right angles from the axis and are small, membranous, brown colored structures. A microsporophyll consists of a stalk with a terminal leafy expansion, the tip of which is projected upwards and is known as apophysis. 2 pouch-like microsporangia are present on the abaxial side of each microsporophyll. In each microsporangium are present many microspores or the pollen grains. Each microspore is round and yellow colored, light, uninucleate with 2 outer covering, that is, outer thick exine and inner thin intine. The outer exine protrudes out on 2 sides in form of 2 balloon-shaped wings which help in dispersal of pollen grain. A few microsporophylls of lower side of cone are sterile. Coming to the longitudinal section of female cone. In the center is present a cone axis and many megasporophylls are arranged spirally on the cone axis. A few megasporophylls present at the base and the apex of the apex are sterile. Megasporophylls present in the middle of the stabili are very large and they decrease in size towards the base and apex. Each megasporophyll consists of two types of scales known as bracts scale and ovuliferous scale. The bracts scales are thin, dry, membranous, brown colored structures having fringed upper part. These are also called as carpillary scales. An ovuliferous scale is present on the upper surface of each bract scale. Each ovuliferous scale is woody, bigger and stouter than the bract scale, and it is triangular in shape. A broad sterile structure with pointed tip is present at the apex of these scales, and this is known as apophysis. At the base of each upper surface of ovuliferous scales are present two sessile and naked ovules. The micropyle of each ovule faces towards the cone axis. Each ovule is anatropous and it remains surrounded by a single integument consisting of outer fleshy, middle stony and inner fleshy layer. It opens with a mouth called micropyle. Integument surrounds the megasporangium or nucellus. Just opposite the micropyle is present a pollen chamber. Nucellus encloses female gametophyte which bear 2 to 5 archegonia at the micropylar region. Ileus of seed. The seed is enveloped by a seed coat developed from middle stony layer of ovule. Inner fleshy layer may survive in form of thin membrane. Outer fleshy layer disappears. A thin membranous and papery structure called perisperm develops inner to the seed coat. Well-developed endosperm is present. In the center is present the embryo consisting of hypocotyl, radical, plumule and 2 to 14 or more cotyledons. Thank you.